So hello everyone, and thank you so much for coming here. I have probably met a number of you. Those of you who are not students in the medical program, I probably haven't met yet. But I'm very happy that you've taken your free lunch hour to come here and listen to whatever I'm about to talk about, which is something that I'm personally very excited about, and that's the development of a type of clinical research that I predict will be very, very successful and hopefully very important in the coming 10 years, 20 years, who knows. And so I've called my presentation Gold Digging in Registries and Biobanks. So I predict that there will be a golden age. You know, in our society, I've been told by people who are really clever that there are two opposing traditions, and one of them is to look back on the paradise that we have lost. That in the past somewhere everything was great, and nowadays things are not as fine. And you of course recognize the myth of the Garden of Eden, the myth of the sunken Atlantis, and the lost Avalon of King Arthur, etc. On the other hand, we also have the modern times with a very optimistic feeling of an development, of an evolution of society, nature, and technology. And there also in religion we have aspects of something to come which is greater than today, where everything is just just misery. When it comes to clinical research, for a number of years we have been thinking that the Swedish medical research, particularly the clinical research, is in big problems. And there have been lots of articles written about this, lots of statistics indicating that things are not as great as they should be. One example here, another example here from a funding agency. So we tend to look back on a golden age where, when things were much better than today. Here in the Karolinska, of course, we have our own mythology. We look back on the times when Seldinge in 1953 discovered, or introduced, I should say, a catheter that was very, very key to the development of X-ray angiography of blood vessels. Another famous example also from the 50s is the development of the pacemaker. The first pacemaker, intracardial pacemaker, was introduced here in the Karolinska by these famous people. And of course, nothing can be as good as it used to be. So there is a crisis in clinical research. But the message in my presentation today, and the thing I would like to convey to you, is the feeling that we have a golden opportunity. We're standing in front of a virtual gold mine when it comes to clinical research, with an impact on medical research in general. There was a book written a couple of years ago, was called Guldgruvan I hälso- och sjukvården, the gold mine in healthcare, which focused on the so-called quality registries, kvalitetsregister, indicating that this would be, or is, an underused resource. Registries. I mean, registries, it's not a very sexy word, is it? I mean, you tend to think about very boring gränssnitt. Tend to think about files, meters and meters of files. And I will try to get you a little bit of a different opinion by the end of this presentation, I hope. We have a special asset here in this country. We have a catalog of about 100 kvalitetsregister, quality registries. This is the homepage which they have for these registries on the homepage of SQL. I will come back to what that is. So the quality registers, they have a purpose of quality assurance of healthcare, at the same time to make sure that healthcare services are evenly distributed and that results of healthcare are also evenly distributed across the country. But they have also the purpose of being the basis for research. These are all funded publicly by SQL, Sveriges kommuner och landsting, and Socialstyrelsen, the National Board of Health and Welfare. But they have all been funded by enthusiasts, eldsjälar, and they are still run by the profession. So it's nothing that the authorities invented and told us to do. This is something that came from the grassroots upwards. These are quite unique. There are no other countries where they have such a long catalogue of quality registries. And this gives us a special asset in this country to do research based on these. These are four of the more famous ones: the Swedeheart, the protesregistret, höftprotesregistret, the registry for rheumatic disorders, and the intensive care registry. Then hopefully there is some gold there, and I will try to explain why there is gold to be found. Personally, I've been engaged in the MS registry, multiple sclerosis. And what is multiple sclerosis? Well, those of you who listen to my lectures on the first weeks of the medical program may remember that, or may have seen these figures before. MS is a disease of the central nervous system, the brain and the spinal cord, and it's characterized by inflammation. And on MRI, magnetic resonance imaging, there are white spots that come and go with time. If I press the button here, you will see these dots coming. This doesn't happen in real time, of course. This is over the span of 2 years, sequential scans of the same slice of this patient's brain. Things that come and go. On the same time, on the other hand, there's another process, and that is one of loss of a brain tissue. Whoops. I need to be very precise here, I think. So this is an extreme example. But in addition to these focal spots of inflammation, there is also a general loss of a brain tissue. This is a patient that had a bad case of MS, and this loss of brain tissue was over the course of 8 years. So it's nothing that happens fast. Of course, these focal inflammatory lesions, they are caused by various kinds of blood cells, lymphocytes, macrophages, and others. And mainly they target the myelin, the sheaths that around that enclose or embrace the axons in order to make them propagate signals faster. When that happens, it's not always so that only the myelin is targeted or is damaged. In this lower right corner here, you see the little green dots here, and those are swellings on nerve fibers that have been cut off. After cutting off, there would be a swelling like that. So there's also a certain amount of irreversible damage. And hopefully, or we tend to believe that the focal inflammation here is actually what results in this, or what is the reason for this general loss. But it's formally not really proven that that is the case. That's one of the remaining problems in MS research, one could say. In some other way, MS can be depicted in this way. This is a time axis. These are the symptoms that come and go. Each of these things here, when you get a symptom, you have a relapse, skov in Swedish. And then you get better again and you have a remission. And after a couple of relapses, you may not entirely return to the healthy state, but you're left with some kind of disability. What is strange for MS is that after a number of years, in addition to the relapses, you also get a progressive disease, a gradual worsening of disease. It doesn't happen to everyone, but it happens to most people with MS. So it's a chronic, lifelong disease. 1 in like 250 people get MS, and this is actually more than we used to think. We used to think it was 1 in 1,000, but it's actually more common than that. It is caused to some extent by genes, to some extent by environmental factors, and to a great extent by pure bad luck. We do have some disease-modifying drugs. Those are drugs that are actually able to decrease the number of relapses and to make the relapses that come anyway milder. But so far, once the progressive stage has started, we don't have any medications to stop that, and that's the remaining big problem in MS. Okay, so that sets the basis for the rest of account of MS research. So, to be a bit personal, I actually completed my thesis work in 1993. And that was the ugly cover of my boring book. I was very happy though. Looked a lot younger in those days, of course. I was also already then a certified neurologist, so I treated patients with MS. And what happened in those years was that the first drugs came. So I had a certain amount of need to have a registry. I wanted to carry on doing research, and I wanted to use clinical information in my research projects, and I needed to get some kind of order in that. And I couldn't extract data from the medical records because that wasn't allowed and not really practically possible because they were all paper in those days. Typed, but paper. So there was a research need, but there was also a clinical need. I needed to have some kind of a list of the patients that we had put on these treatments. And the medical records, they were just paper. So I had a computer that was, well, not as bad as that one, luckily, but still fairly basic. And we made a very simple database for that. After a couple of years, my boss in those days told me that we had to contact other colleagues. So we started a network and we started to talk to other neurologists treating MS patients in the entire country. And after some time, we had a consensus on what to register and how to do it. So we launched a registry. Today, it looks like this. You can actually see, if you see the lines here, they actually look a little bit like the course that I showed above of the patient, right? So the red dots here, the red line, that is the disability of this patient. Here you have names that I've of course hidden. There are lots of modules down here of various things that you can register if you want. But the most important thing is the disability, the red dots here, and the treatments. Because the main purpose of this registry from the beginning was to evaluate the effect of treatments. And those colored lines here represent the various disease-modifying drugs that specific patient had received or has received. And you can see here that in the beginning things got worse, but then after some time things stabled. And that's very clearly an effect of the medicines, because if the medicines hadn't come here, the normal development would have been of progression like that. So this is very clear evidence for this specific patient that in this case we actually did something good. So every time I get a patient, every time I see my patients, I open this up, I get a summary of all the clinical pieces of information that I need in order to deal with the questions that day. And I even show the patient this. You know, this is where we are, this is what we have done, now we decide to do that because of this. So it comes really handy in from a practical point of view for me as a doctor. So we have this registry now for 13 years, in the sense that from the 1st of January 2001 we had public funding for the first time. Then we've had only public funding. It's available over the internet, so it's actually used now in every department of neurology. It's been web-based since 2004. It's a decision support tool in the way that I explained. And we actually have some information on about 80% of our prevalent patients in this registry, which is very good. There are very few other registries in the world that come to a higher degree, to a higher frequency of coverage. The Danish registry is better. They have 95%, but they have much less information on their patients. So I still think we're doing good. And we use this for various scientific purposes, epidemiology, post-marketing follow-up, creation of biobanks. And to date, we've actually based almost 90 scientific publications on the registry, to some extent at least. So The development has been quite favorable. These are the number of patients in the registry, so we add like 1,000 patients a year. Not quite in the last few years. We may be reaching the ceiling, so we probably have some leveling off. So it works very well, and this may be surprising because this registry is voluntary. The colleagues use this because they want to. And why do they want to? Well, they want to use it because they get some kind of information back. I think the basic argument is, of course, that they get this interface back. It's practically very useful to have this registry. First time, of course, when you have a new patient, you don't have much use of it. But after some time, after a couple of years for each patient, it's really, really handy. In addition, we also provide a data analysis tool. We can extract data on the patients, my department's patients, or my county's patient, and on the national level also, available in some preformed graphs that I can extract from the register when I want. And if I want to do some research personally on my patients, if I have the permission to, then I can export into Excel format and take that further into some statistics program. And of course, research projects can also be performed on the entire registry or parts of the registry. So we have a number of nationwide efforts based on the registry. And this is important because these three specific projects here, the GEMS, the IMSE, and the AIMS projects, they have allowed us to take the research to another level. These are patients that have been invited. Either the AIMS patients were so-called incident MS patients. That would mean patients that are introduced as they are being diagnosed. So just new patients are accepted in that specific study. In the GEM study, all patients are welcome. In the IMSE study, patients on the last generation, highly efficacious MS drugs are invited to participate. And I would say that the vast majority of patients that are asked say yes. I've had 2 patients that declined to take part in the registry, and one of them came back and changed his mind. So for all these patients now we have genetics, because we ask them to hand in a DNA sample, a blood sample. We have serum or plasma. We have asked them to fill in questionnaires that look like that on their behavior, on their lifestyle habits, background, the things they eat, the educations they've had, infections they've had, how much they smoke, how much they exercise, and so on. So we have genes, we have environment on all these patients. And the nice thing, and now I have those people up here, is that we have now close to 9,000 patients in these projects in the biobank. They've already been genotyped. Their DNA has been analyzed extensively. We have information on lifestyle. And now we can connect this information with the clinical information in the registry. There are centers out there in the world that have almost as many DNA samples of MS patients, but none of them have bothered also to collect lifestyle and exposure. And I'm boasting a little bit here, but I'm enthusiastic about this, and so take this as my subjective account of the situation. And none of them have also collected lifestyle, so we are fairly alone in being able to study the interaction between genes and environment. And now we can also add the clinical data to that. So we can study the genetics of treatment response, the genetics of disability, the importance of lifestyle on disability, or lifestyle on treatment effectiveness. So we can connect these in a very unique way, which wouldn't have been possible unless we had this registry. Now we live in a country which is ideally suited and organized for epidemiology. And that's because we have these fantastic public registries. We have from Försäkringskassan Health Insurance Registry. We have at Socialstyrelsen a National Patient Diagnosis Registry, so we can look at comorbidities. We have a big database for all prescribed drugs. Remarkable. We have lots of information on each and every one of us in the population in SCB, Statistics Sweden. And there are, of course, as you know, a multitude of other registries, which we can link these data to. So we send in a file of patients in the MS registry. We get back an anonymized file with a combination that we can then analyze. And all this is being possible because of the personal number, personnumret, because every individual has the same kind of identifier in all systems, which may sound as something trivial, but it's just available in like half of Western European countries and in a few other countries. So this allows us to do quite a bit of different things. So far, we have identified genes, we have identified environmental factors, we have studied gene-environment interaction, we have surveilled MS treatment, novel treatments, we have assessed the long-term effectiveness of treatments, we have identified the effects and also the frequency and so on of anti-treatment antibodies. We have widely studied comorbidities in MS, the occurrence of other problems. And we are now heavily going into health economy aspects also. So just to have very few examples here. When I wrote my thesis, when I started my thesis, there was one gene known, which was an MS risk gene. When I completed my thesis, the number was still 1. It took another 10 or 15 years until we made any kind of progress. And that was in 2007. But now, going from 1 in a number of years, few years, we've actually now exceeded 200. This needs to be updated. So we have identified 200 genes. We didn't do that all on our own, but we did that together with other centers in the world collecting big numbers of DNA samples and controls. So we've really solved the problem. So now we have more than 200 MS genes. We have studied a number of environmental exposures that I will not go into in detail, apart from smoking and snus, oral tobacco. But there are now between 5 and 10 environmental risk factors that are actually true, something we didn't have some years ago. So how about smoking? Well, smoking has been studied by a number of different groups. And this slide is not very nice because you can't see much, but you can see here that odds ratios, that is the risk contributed by smoking. So if you ever smoked, you have 50% greater chance of getting MS. If you're a male smoker, ever smoker, it's 80% increased risk. On the other hand, if you use snus, you can actually counteract this. And this is the odds ratio again. So the longer you snus, the lesser the risk of MS. And this is a bit interesting because it indicates that it's not the nicotine that gives you the risk, it's something else. And we don't really know what it is yet, but it appears that it's actually something that has to do with irritation in the lungs. Which is like a novel concept. One may see that, this may seem trivial, but this is a true observation. It's been observed, it's been confirmed, and it really points towards a mechanism that is now being investigated. So another dimension is then to combine the genes with the environment. So the blue things here, columns here, they show you the increased risk in presence of the genes you don't want to have if you don't want to have MS. But if you're a smoker, this genetic risk is augmented, is amplified several times. So there's interaction between smoking and the genes, which is a bit interesting because it indicates that smoking and those genes actually somehow interact in the functional aspects. So that also gives some clue on the underlying mechanisms, really. Well, there are other risk factors. Epstein-Barr virus, you may have heard about. Practically no patients with MS are negative for antibodies against EBV. On the other hand, 98% of the rest of us are also positive. But patients with MS, they have higher titers. And the higher you have, the higher the risk of MS. You can also integrate that in this scheme. So if you combine smoking, EBV titers, and the genetic factors, the basic ones, you really get a risk stratification from 1 to over 30. And then you may approach a level where you can actually understand why a certain person in 250 gets the disease. Well, so looking back again, when I presented my thesis 20 years ago, that one genetic risk factor that we already knew about was the only factor we knew about. Now we have 200 plus MS genes, and we have a dozen or so environmental factors. And this is a fantastic development. Well, so these are all the risk factors, and most of them are just barely above 1, meaning that they all confer just a tiny little risk. If you look at the top ones, I'm sorry for the colors here not coming out very well. The strongest ones are, well, the blue lines here are HLA-related genes. The pink ones are environmental factors. So the environmental factors that we've been studying here are actually quite relatively important. The stars with the darkest pink are the ones that we have identified ourselves with the help of our registries. The lighter the pink, the more other centers have contributed. But practically, by being members in the genetic consortia, we contributed to all of these, more or less. And that's quite satisfying. We've done our share for this tremendous success. So why do we have success? Well, epidemiology has been around forever, but in the old days people used old methodology. So technology has developed, but the main reason is actually, that we have increased numbers. When I did my MS work in the beginning, I'm sorry, I lose part of the text here. We had typically 50 to 200 patients and as many controls in the study. 10 years ago when things started to go well, we had some 3,000 patients and controls in every study. Now we have 25,000 patients and controls in the studies when we can have that. And that is the main reason why we have made all these discoveries. It's not necessarily technology. This is a modern gold mine. It's quite an impressive technological achievement also, of course. So, what is the way ahead? Well, my own research group focuses on using this integrated data. And we think that by combining baseline information like genetics with exposures acting on the course of the MS patient, like treatment, like lifestyle, like exercise, like, I don't know, sunshine, we have outcomes. And we need to improve the baseline knowledge, we need to improve the exposures, and we need to improve the outcome information. So we're working on all these stages, but it's mainly the same kind of system. And that could not be done without a model like that, which actually puts us in a very unique situation. So actually, to have this longitudinal perspective of a patient's course is actually what is needed for a number of key problems in MS research, as well as in many other clinical disease research fields, of course. So if we want to identify the driving disease mechanisms, we need to assess the course, of course. If we want to have biomarkers and validate them, then we need to have a long course to validate the outcome on. If we want to study subgroups, identify maybe diseases that shouldn't really be part of MS because they are different, then we need this longitudinal perspective. If we want to know whether these drugs that we use are really good over 10 or 20 years, we need to look in the long term. If we want to tell our patients to stop smoking, then we need to prove that over the long term. Otherwise, we couldn't really say we know. So, I think that we have the second golden age for MS, for clinical research in medicine in this country. And we will have that if we improve the registries, if we structure the clinical work in a way that makes it possible for us to document what happens to patients, if we collect the data systematically, if we improve the biobanks and really use the biobanks that are out there. A colleague of mine just discovered in the biobanks that we have samples from MS patients, from 1,000 MS patients taken before they got MS in various biobanks in the country. A fantastic asset, you know, to study the disease before it's even started. That's fabulous. We need to use the high throughput omics techniques of, for instance, SciLife Labs, and we need to feed back what we learn here to the basic researchers, because these are pieces of information that they should try to decipher. They should try to disentangle the mechanism. That's not for us clinicians to do. Authorities need to support population-based research, and authorities do that here. The quality registries get lots of support. If we do this, we will find the causes of diseases, like in the example here, and this is the coming kingdom then. We will identify driving mechanisms, identify and validate biomarkers, disease entities, new therapeutic targets, identify consequences of disease for patients themselves and as for society, of course, and evaluate outcome of what we do patients also. This is a little bit technological. This is bibliometry. This is my own clinical department of neurology. And this is the global average. And in 2002, we were very average. In the last 10 years, we have emphasized research on registries, population-based research in big numbers. And we've gone from average to excellent, purely by concentrating on population-based research. This is a way of assessing what we need to do. If we have registries in the middle, we can effectively study biobanks, animal models, genetics, mechanisms, and develop new drugs. If we evaluate what healthcare provides to the patients, society will return investments in healthcare. And if we are really good in the middle here, both wheels will spin faster. So to my mind, this is indeed the new gold mine. And it shouldn't say keep out, it should say welcome. There's plenty of room for you guys to do research with these tools in the future. And that will be my last words, yes. We're happy to take questions. Thank you.